Secretary, the Principal Sir and Madam Radhika for having invited me for this talk. As Dr. Radhika mentioned, I have been associated with the V.O.C. College since 2002. It was in the year 2003 I first made my maiden visit to V.O.C. College. I talked to the students and staff there. And even though I did not visit after that, but we still have a very good correspondence and communication through emails and the social media. Thank you, Madam and the Principal, for having invited me. And I also welcome all the participants on behalf of myself. I realized there are people from many parts of the India as well as several overseas participants. I welcome you all on my own behalf. I also realized that, understand that there are 50 or 60% of the participants are bachelor's and master's students and research scholars, and some of you are faculty members. So my talk will cover some of the basic aspects so that the students can get benefit. And also at the end of the session, I will be happy to take your questions through make it interactive session. Before we move on, as Dr. Radhika Madam mentioned, I have been working in several universities after my PhD, United States as well as in Singapore, researching on malaria. But I finally moved to Temasek Polytechnic because my mind was in teaching. So I like to be involved in teaching and interact with the students. So sharing the knowledge give the much happiness. So I decided to move on to the teaching, but still I do a little bit of research. Temasek Polytechnic is one of the 5 polytechnics under the Ministry of Education, Government of Singapore. We have 1,400 staff and nearly 15,000 full-time students, and we have collaboration all over the world. And specifically, I belong to the department or School of Applied Science, and I teach medical biotechnology. Our students basically, after the graduation, they work in the medical or clinical or research and diagnostic laboratories as technologists. More recently, many of your students are involved in COVID-19 diagnostic because of the surge in COVID-19. These are some of my students from Open House, and I have also one student, Darren Chua. He is a Asian Games medalist and going to participate in Olympic Games as well soon, representing Singapore. So moving on to my talk, I know some of you are from Uttar Pradesh. I hope some of you, I am sorry for the background noise. I am working from home and there is a air base as well as a train station nearby. So bear with me for the background noise. So I know at least some of you know this gentleman. He is a freedom fighter. He was the Chief Minister of Uttar Pradesh during Indira Gandhi period. He was also Chief Minister of Uttarakhand. He has the fame of being the Chief Minister of 2 states, and he served in several capacities. What happened in early 2000? A guy named Rohit Sharma, this guy claimed that N.D. Tiwari, I have very high respect for this man, and he is actually a freedom fighter. So Rohit Sharma claimed that N.D. Tiwari was his father, and N.D. Tiwari was Narayan that Tiwari was refuting it. So Rohit Sharma had to go to the court and ask for the DNA test. He went to then finally the Delhi High Court ordered N.D. Tiwari to give the blood sample, and the blood sample, the results came positive. And finally accepted that Rohit Sharma was his son. And happy ending, N.D. Tiwari also married his mother at the age of 82 years. Unfortunately, this Rohit Sharma was killed by his own wife 2 years ago. That is a sad ending. Just to give you an highlight, because the DNA fingerprinting is very common in India since 1990, but many people know of this DNA testing or DNA fingerprinting when a very famous personality like N.D. Tiwari was involved in this case. It is the idea of giving this background information. So I just mentioned this. These are all available in the data. I mean, newspaper clips and all. So let me move on to the topic. We all know, most of you are biologists, so we all know the human genome contains 3 billion bases, 3.3 billion bases exactly, and about 20,000 genes on 23 pairs of chromosomes. So about 2% of the genome codes for protein. Only 2% of the genome of this 3 billion bases codes for protein. What about the rest of the genome, the remaining 98%? Earlier, we used to call them as junk DNA, but no longer we call it junk DNA. Rather, we call it functional elements. They are the one controlling the remaining 2% of the expression, cell cycle regulation, and even including the cancer, everything depends on these functional elements. So what are tandem repeats? As the name indicates, repeats, this DNA, certain DNA sequences, they repeat one after another in the genome, in the human genome. So they are called tandem repeats, and they are widespread throughout the human genome. So it is present all over in all the chromosome, and they account for 3% of the human genome. And the interesting thing is they show sufficient variability among the individuals. That means the number of repeats that you and me have different. That is what makes the basic for this DNA fingerprinting or DNA profiling. These tandem repeats, in case I'm going fast, please let me know. So the tandem repeats are classified according to the size of the repeat, how many bases in the repeat, either it is a 6 base or 10 base or 100 base like that. And also how many times this bases is repeated. So they are classified based on this into 3 categories, namely microsatellites, mini satellite, and macrosatellite. So the microsatellite has about around 10 bases, and the mini satellites 10 to 100 bases, and macrosatellite about 100 bases or more than 100 bases. Why they are called satellite? Because of the repetitive sequences. When you separate the genome by density gradient centrifugation, these repetitive sequences form a separate band or a satellite separate from the main genome. That is why they are called as satellites, mini satellites. So the mini satellites is also called as variable number tandem repeats. As I have just mentioned, they have the nucleotide number of 10 to 100 bases depending on the locus. So the earliest DNA fingerprinting was discovered in the early 80s by Alex Jeffrey. The methodology he used was basically he takes the whole genome and digests with a certain restriction enzyme. Southern blot and restriction enzymes were discovered earlier before the PCR and all this technology were developed. So he took the whole genome accidentally. He was doing some southern blot and he noticed a specific pattern. So how is it done? The genome is digested, then run on the electrophoresis, then they are
right, sir. Sorry for the interruption, sir. Right, sir.
Probe for the VSTR, variable
sir,
number.
Right, sir.
Yeah.
We cannot see your video, sir. Video on Madlama. Video we cannot see,
sir.
sir.
You can do that, right?
Just try, sir, again, once again.
Yeah, I
yes.
started my video. That is, I think, from my side. I hope my slides are still on now.
Yes, sir.
Yeah,
it's
slides
going.
are okay, sir. Okay, sir. And voice also can be more, sir. You can be more audible, sir.
Okay.
You mean
low.
A little bit louder?
Your voice. Yeah.
Little bit louder, sir. That's enough.
Okay, sure. Yeah, thank you. So how this is done earlier days, you take the whole genome, digest with a restriction enzyme, then you run on the gel, then you transfer to the nitrocellulose membrane. Because when you digest the 23 pairs of chromosome, 3.3 billion bases, you have thousands of bands. You don't know where is VNTR, where is STR and all. So you need to probe with the VNTR, auto radio, radio labeled probe. Then you go for X-ray analysis. This is very cumbersome. This is the discovery made by Alex Jeffrey. Okay. So look at this picture. Let us take one VNTR as an example, which is DS180. It is in the non-coding sequence. The repeat has 16 nucleotides, and the longest repeats are 442. So that means in our genome, we can have either one repeat, two repeat, three repeats, or up to 42 repeats. If you have, we don't know, we have two copies of DNA, right? One from father and one from mother. So that is why you see two different bands here. Okay. So the one repeat from is from one set of DNA, the other one is the other copy of the DNA. In some cases, you will see only one copy, one DNA bands. That means both chromosomes have same number of repeats. Okay. So this is very, very rare. About 50 to 20% of the population have the homozygous expression, homozygous nature of this STR or VNTR. So this is the classical way the DNA profiling or fingerprinting. Earlier, Alex Jeffrey called it as genetic fingerprinting. Okay, it was done in the earlier days. I will come back to the modern technique. It is done later. Before, I would like to explain what are microsatellite. Microsatellite, as just showed you from the previous speaker, is less than 10 number of bases. And they are repeated about 5 to 3 times in the genome. They are also called as symbol sequence repeats. Symbol sequence repeats. Because you have only 10 bases maximum, they are repeated 30 times only maximum. Even if you amplify, the maximum number of PCR product will be 300 bases. So it is easy to digest, easy to amplify compared to the VNTR. So the the microsatellites or STR are also show substantial polymorphism and are abundant, very abundant. Every 10,000 or 15,000 bases in the human genome, you will see one STR. So these are suitable for analyzing forensic specimen, because when you get the forensic specimen from the crime scene, most of the time, because the DNA or sample is exposed to the harsh condition like high temperature, humidity, or even sometimes it is a rainy day, and sometimes the DNA is from the body or sometimes from the floor and the instruments used for the killing, murder. So the DNA is mostly will be degraded. Because these fragments are shorter, they don't undergo much degradation. So this can be amplified efficiently. So that's just to give you an idea. So I told you we get one copy of the chromosome from one set of chromosomes from father and another set from the mother. So when you have the DNA from the mother and child, there are two male here. We need to establish who is the father of the child. This is a very simple cartoon picture from the McGraw-Hill that we use for our teaching purpose. So we don't have any color coded when in the radiograph or in the gel. So you can look. So these bands from the child comes from mother. This band comes from mother. These two come from male one, and this also comes from the male one, and none of the bands is from male two. So we can easily say that male one is the father of this child. But it is not as easy as we see from the picture. There are a lot of statistical analysis, a lot of complex algorithm are involved in order to make a conclusion. So we will see all this in detail. I am just giving you an overview. So we talked about the VNTR and the STR, and here is the timeline, the significant developments in the impacting that made a major impact in forensic genetic analysis. So as I mentioned, restriction enzymes were discovered in 1970 or 71. Southern blot was discovered in 1975. Name of the scientist, Edward Southern. So that is why it is called the Southern blot. Western blot and Northern blot are just jargon. So Southern blot is a hybridization of DNA and DNA. So VNTR was discovered by Wyman and White in the year 1980. Later comes PCR. Karim is discovered, invent PCR. The same period, Alex Jeffrey in UK, he accidentally find a specific pattern when he was analyzing the genome of the family members from his own lab. Then he understood that he can make this tool to identify the people from their own genome. So he was actually doing the autoradiograph. I told you, right? You take the whole genome, digest, transfer to the membrane, then use the probe, then you take the autoradiograph. It is a very complex process. But the PCR was discovered during the same time. Then we also discovered the STR, the human genome sequences were starting up. So the people thought that why not we use STR, because they also show substantial polymorphism. And when you use the RFLP, restriction fragment length polymorphism, you need plenty of DNA, several microgram of DNA. But from forensic crime scene, you are going to get little bit of DNA from the hair or tissue or blood or saliva or semen or some tissue, saliva, whatever it is, just small piece of sample from the culprit, right? So this is very useful. And DS180 is a major, one of the very common VNT used for analysis by many of the countries. And in the US, the offender DNA database was established in the year 1997, and the real-time PCR came later. So this is the timeline. You can see how the VNTR was discovered. Subsequently, the RFLP, then comes the PCR, and technology changed from RFLP to PCR-based technology. You can call it AFLP, amplified fragment length polymorphism, but AFLP is slightly different. Okay, that is not now usually used for the plant genome analysis. So in both cases, amplification is involved. So I'm just going to talk about some information, give you some information about CODIS database, DNA database. I think I am going a little bit faster. I will slow down my pace. So the CODIS stands for Combined DNA Index System, which is nothing but a software developed by the FBI, Federal Bureau of Investigation, similar to our CBI in India. Okay, so this is a software, and there is an organization called National DNA Index, which actually stores all the data, DNA data from the US citizens, which accounts for nearly 4% of the population. The US population is currently approximately 320 million, 32 crores, and there are 1.4 crore people's DNA already deposited. They include offenders. That could be also even just simple traffic offense. Okay, that could be also, it is not like a criminal offense. So offender, then arrestee profiles, and also forensic profile. That is a DNA profiles obtained from the crime scene. They have separate database. So in the as on April 2020, CODIS has produced 511,000 hits, assisting nearly 500,000 cases or 5 lakhs cases in investigation. So what are the CODIS core loci? So we talked about one of the very common loci. I mentioned DS180, but with one loci, we cannot make any meaningful information. I told you this is a very STR are very common among the human genome. Every 15 or 20,000 bases, you find one STR. So this CODIS uses 20 different loci. Before 2017, they were using only 13,000 loci or 13 STR. But from January 2017, they added 7 more STR. So they have 20 loci. And remember, these 20 different STR have multiple alleles. That means in you, it may be like four repeats. Let us take example of fibrinogen alpha. It is actually a gene coding for the fibrinogen involved in the clotting, blood clotting. Let us take fibrinogen. The fibrinogen may contain 1 repeat or 2 repeat or 3 repeat. So they have about 29 alleles, 29 different form. In my genome, I have 2 copies of the DNA. One of my chromosome may have 10 copies. Another chromosome may have 10 repeat, 10 copies, 10 repeats. Another chromosome has 12 repeat. In you, it is different. So each target STR on average have 20 or 25 different alleles. So you have 2 copies of the DNA in your genome. We are going to analyze 20 STR. That means we have 40 copies of the template, right? We have 40 templates. On average, let us assume that we have each STR has 20 or 25 alleles, okay? 25 different versions in DNA. That means by employing this technology, we can have 40 billion combination. That means that is the total population of the earth is 8 billions. Okay, if you add one or two more STR, that is going to be in trillion. So the probability of two person having the same profile is very, very remote. That is what make it very specific, similar to your fingerprint. Fingerprints are different. It is like specific to every individual. Similarly, the DNA pattern is also specific. That is why it is called as DNA fingerprinting. I'm going to give, probably some of the students may not have some, still not clear about the repeats and loci and all. I'm going to give you more information on this STR. I'm sorry. So I mentioned there are 20 loci that the FBI uses for their investigation, forensic investigation. For teaching purpose, I know there are many of you are teachers, lecturers, and working in colleges. For your information, we also do the DNA fingerprinting in our laboratory for teaching purpose, but we don't use all the 20 loci for the teaching purpose. The idea is to explain the concept and application to the students. We are not dealing with any forensic sample as such, or sample to determine the paternity. So we use only 3 loci. One is called fibrinogen alpha, which is also called as fibra, which is localized or located on chromosome number 4. It has 21 different allele. Okay, that means how many repeats you have, 4 or 5 or 6 and so on. So, and these alleles vary in size depending on the number of repeats they have. Another one we just talked about, D1SAT, which is located on, I think I'm still going fast. Let me lower down. So another one is located on chromosome number 1. There are 29 different alleles. So the repeat is 16 bases, which is basically a VNTR. We not call STR. So it is almost similar to the number of bases are lower. You can call STR, VNTR, doesn't matter. So either you can have repeat of 1 repeat, 2 repeat, up to 42 repeats. So you can imagine how many alleles are there in the D1 SAT. Apart from these two, we also use another marker, which is actually a gene coding for amylogenin. Okay, it is actually a sex gene which is located on both X chromosome and Y chromosome. In X chromosome, it has about 1000 bases. In Y chromosome, it has a deletion. 200 bases are deleted. So it has only 800 bases. So what happens when you do a PCR? The sample is from female, you will have only one band, because in female you have only X and X. That will give only one band at the size of 977 bases. If you have a sample from male, you are going to have two bands, because males have X and Y. From X chromosome, you are going to have a band 1000 base pair. From Y chromosome, you are going to have a band 800 bases. These two bands will be close to each other. When you resolve in the gel with the 0.8% gel or 1% gel, you will be clearly able to distinguish the two bands, and you will be able to make a conclusion whether the sample is for a male or female. There are some issue because this gene may be deleted in the Y chromosome, so there are better methods to determine the sex of the sample. So let me give you one more example about the repeats. The students may be still confused. And imagine, so far we talked about the non-coding sequences. Now I talk about a coding gene, FGA. If you have 4 repeats, if I have 5 repeats, if someone else has six repeats, it is not going to code a meaningful protein, right? The protein will be mutated. That fibrinogen will not be active protein. It will not be helping to the blood clotting. But fortunately, this STR is located in the introns of the gene, FGR gene. So if you look at here, there are 21 alleles possible. Either 1 repeat, 2 repeat, 3 repeats, 4 repeats, and so on. Either you are going to have 2 repeats in one of the chromosome, in another chromosome you are going to have 2 or 3 repeats. Not all these 21 repeats in your genome. So you are going to have only 2. Sometimes In both the copies, you may have same number of repeats. That is called homozygous. Here is your forward primer. Here is your reverse primer. When you amplify, you will be still able to differentiate even the 4 bases. By normal electrophoresis, you will not be able to distinguish, but we have advanced technology now that I will discuss later. So I mentioned earlier, so because there are many students here, I will be a little bit going through the basic, and also I have designed this lecture because there are plenty of teaching staff. They can also develop a similar experiment for their teaching purpose. So the experimental flow is simple like this. Of course, we need to extract the DNA first. Then we do the DNA fingerprinting, which is nothing but a PCR. In PCR, you just use one set of primer to amplify something, insulin or fibrinogen. You have forward primer, you have reverse primer. But here, remember, you are going to use 20 different primer or 20 pairs of primer, right? 20 sets of primer. It's a very complex PCR. You need to optimize the melting point, the salt concentration and all, because these are all carried out developed by the FBI. You can imagine how much funding and how many number of scientists have involved in developing this 20-plex assay. So the second part is the multiplex PCR, what we call as DNA fingerprinting. It is basically a multiplex PCR. Either in the laboratory when you are doing, you can use a normal PCR machine. If you are just doing 1 or 2 target, you can do a normal PCR machine. And normally for the forensic purpose, they use the capillary electrophoresis nowadays. No, they are not doing any like RFLP autoradiography, just capillary electrophoresis that will give you how many repeats, and also if there are 1 or 2 bases extra, that will give you 13.2, 13.4 and so on. The last step is electrophoresis. That is, in our case, for teaching purpose, we do the normal gel electrophoresis. But as I just mentioned, the NDIS or the laboratories involved in paternity tests or forensic analysis, they do capillary electrophoresis. And finally, we analyze. It is not done by manually, as I shown in the earlier picture. It involves a software, a lot of software I will show you later. So they do a lot of mathematical calculation, which involves a lot of algorithm, because this is not just like a test like what you call a simple test, like a uric acid test, whether you say positive or negative. These are all some life-changing decision, okay, from paternity test. If you find your son is not your actual son, so we cannot depend on some simple test. So it involves a lot of mathematical calculation and statistical analysis used offered by the algorithms. So let us move on to this DNA extraction part. I have made it some animation because there are a lot of bachelor students here, about 200, 300 students. So what you see for DNA extraction, you can use any kind of tissue from your body. The most common way, nowadays, the COVID-19 diagnosis, they all take the sample from your nasal swab or oral swab. That is what we use for our laboratory, and the code is also used, suggest the similar methodology. But if you are going to have the sample from crime scene, you are going to have a tough time, because maybe you have a saliva or semen or blood or piece of tissue, hair, nail. Even if you don't have the DNA, the weapons they use, they can also get from the knife handle and all, or the clothes. Anything can be the source of DNA. For teaching purpose, we use the cheek cells. These are my own cheek cells we do in the cell biology lab. You take the toothpick, just scrub a few times and make a gentle smear on the slide, put a ethylene blue or toluidine blue, and then you can put a cover slip and look under the microscope. You can see the beautiful cheek cell. There are 2 cells here, nucleus, and the membrane. It is a acidic stain. So all the acid components of your cells are beautifully stained. So we take the and we don't take the toothpick for this purpose, DNA extraction, but we use a specialized brush called cytology brush, okay, which is similar to the brush that you use for washing the bottle, circular. It has the brushes all the side, right? But it is very tiny. So you put inside the mouth, scrap a few times, take it, So you have now extracted the cells. There are only about like thousands of cells, few thousand cells, but that is good enough. If you have even 1,000 copy of the DNA, then when you amplify, you are going to have millions and billions of copies through PCR, right? So after the, so for easy understanding of the student, there are 4 steps for DNA extraction. Okay, so when you break open a cell, you are not only going to have DNA, you are going to have plenty of RNA. DNA, you have only 2 copies in a cell, but RNA you have several thousand copies, or sometimes several million copies, right? Because depending on how much protein you need, the copy number of mRNA is made, multiple transcription and so on. Then you have plenty of proteins, right? Thousands of protein, structural protein, enzymatic protein, and so on. Besides, you also have a lot of carbohydrates, like a lot of multiple carbohydrates. And we need to get rid of all this thing. We need the clean DNA. So it involves 4 steps. First is break open the cell, which we call it lysis. We can use like some acid lysis or alkali lysis or enzymes or mechanical way. The second part is removal of RNA, because your mRNA is also complementary to the DNA strand. So when you are doing the PCR, if you don't remove the mRNA, they bind to the template, thereby stopping or inhibiting the binding of the primer. Then you need to also remove the proteins, right? Because if there is plenty of protein, it is also going to interfere with your downstream processing. So lysis, break open the cell, then remove the RNA, remove the protein, and finally precipitate the DNA and wash it. So this is here, this is what you need. You need the cytology brush and the pipettes and pipette tips, some centrifuge and heat block. That is it. It's a very, very simple requirement. You can develop your own reagent in your laboratory. For lysis, it is sodium hydroxide, sodium sulfate. For precipitation, it is basically ammonium sulfate and isopropanol. Very simple. You don't need to buy any kit. That is the purpose I put the list here. You can also buy commercially if you have the budget for your teaching lab. So in the first step, you are just going to scrap the buccal cells and you put in the nucleic acid lysis solution and transfer to the heating block. In the heating block, it is going to digest. Okay, it is going to lyse your cell and release your DNA, RNA, protein, everything in there. In the next step, you need to cool the tube because you are boiling it, right? If you add the RNase enzyme immediately, the RNase basically is a protein, it will be denatured. Cool the tube, then add the RNase enzyme and leave it in 37 for about 5 or 10 minutes. RNases are very robust enzyme, so they will rapidly degrade all your RNases. Excuse me. So now you add protein precipitation solution. You have the DNA, you have the RNA already fragmented. So the protein precipitation solution has sodium rhodocyl sulfate, which is nothing but soap. It also has ammonium sulfate and many other salt. So they precipitate the protein. You put in the ice, so it enhances the precipitation of proteins. Then your protein is precipitated. Now you do a centrifugation for about 5 minutes. All your protein is sedimented. In the supernatant, you have the DNA, you have the fragmented RNA, you also have several water-soluble sugars. So we need to get rid of the sugars and RNA and also all the salt. Because in the lysis and protein precipitation, we have used many different kinds of salt. So you are going to use either isopropyl alcohol or 100% ethanol, which will precipitate the DNA, not the fragmented RNA or sugars. So after adding the alcohol or isopropanol, you centrifuge, all your DNA is sedimented, okay, and all your free sugars and fragmented RNA, everything is in the supernatant. From this point onwards, you need a pellet, right? So the pellet is not clean. It may have some contaminants of salt and so on. So you are going to wash with 70% ethanol. Why 70% ethanol? Why not simply buffer or water? Because if you use buffer or water, it will also dissolve your DNA. In the 70% ethanol, you have some water that will dissolve the salt. You have ethanol that will keep your DNA precipitated. So after centrifugation, you pour the supernatant and put the tube upside down and dry the DNA. Then you add some buffer. Okay. Then add a buffer and dissolve it. You have a very clean DNA for your experiment. It can be a PCR or ligation or whatever. This is the basic method. Okay. Or southern blot or whatever. This is the basic method. Sorry, I spent a few minutes here for the benefit of students to understand. Okay, let us move on to the next part. The next part is your actual fingerprinting. Sorry, because I need to be a bit louder, so my throat dries very quickly. Unless in the lecture theater, the system is different. So here I need to be a little louder. Sorry about it. Okay, the second part is multiplex PCR. I told you you are going to use 20 different kinds of primer. You can use the normal PCR machine for your teaching purpose because we are not handling any forensic sample or patient sample for paternity testing. So, or in the diagnostic laboratory, forensic laboratory, they use the capillary electrophoresis. So here, what do you need? Again, for the benefit of students, we don't need any hi-fi equipment. Okay, the only equipment you need is the PCR machine, which is about for 50,000 or 1 lakh rupee, you can buy a decent PCR machine because PCR machine is no longer under the license of Roche. Anybody can make your own PCR machine. I am also making a one PCR machine, which is actually a pocket PCR machine for the shrimp farmers. They can test in the farm. Okay, it is called a microfluidic PCR machine. So these are all very cheap. Don't think these are all very expensive. Okay, so you have these are the things you need. A PCR tube. The tube will be very thin compared to your normal tube that used for other tests in the laboratory, like the microcentrifuge tube, because the tube, the heat need to be transferred to the solution inside. So the PCR tube will be very thin. I just gave every student one tube, so I asked them to press and ask them to break whether they can break. Okay, so it is very thin so that it can easily transfer the heat. And of course, you need a forward and reverse primer. In our case, we have only 3 set of primer: fibrinogen, D1S80, and amylogen in 6 gene. In CODIS, you are going to use 20 different, and in some countries, they are using 22, even 25 loci. So that will be, you will get the trillion copies. I mean, combinations are possible when you increase the more and more loci. Okay, so we have this is again, I'm sorry if there are some professors here, some teaching faculty here. For benefit of the students, I made a little bit simple so they don't get lost during the presentation. This is how we set up the PCR. We set up a 25 microliter reaction in which you have the forward and reverse primer of all the 3 fibrinogen, D1S80, and amylogen in gene that you are going to amplify or STR. Then you put the 2 microliter of the DNA that you have isolated from your cheek cell. Okay. Then make up to 25 microliter with water. The master mix that you are using here contains the buffer and DATPs, deoxynucleotide, ATP, GTP, CTP, and GTP, and also the Taq polymerase. Okay, you are not going to use a normal polymerase because the normal E. coli polymerase or human polymerase will denature when you increase the temperature for denaturation, right? 95 degree. Whereas here we use a specific kind of polymerase called Taq polymerase, which is obtained from a thermophile aquaticus, Thermophilus aquaticus bacteria that lives in the hot spring. So they can withhold the temperature. We also need positive control and negative control. Then we also use a gene only in a tube. We also only sex marker. So we know whether the specimen is from a male or a female. These are for teaching purpose. And this is how we set up the PCR. 95 minutes for 95 degree for 5 minutes, initial denaturation. Then you have 40 cycles of denaturation, annealing, and extension. And finally, you extend for 10 minutes. This is called final extension. And we have in our laboratory, we have 3 different kinds of machine. One is the normal PCR machine, applied biosystem. Another one is Rotargene Q from Coagen. This is a real-time PCR machine. And we also have another machine called Quant Studio. This is actually part of the IIN and genome sequencing machine, wherein you can handle about 384 samples at a time. Here you can use about 72 samples at a time. This one is like a tighter bit, your eyes supply. You can run about 94 samples at a time. So normally for teaching purpose, we use this machine because it is not a real-time PCR. It's a regular PCR that you are going to run on the gel. Then the last part is analysis of your result, right? Analysis of result. As I mentioned, for teaching purpose, we use the electrophoresis. And in the organization like CODIS or CCMB in India, they use all the software, capital electrophoresis and software. This is Professor Jeffrey Alex, who actually discovered this RFLP first. Here you can see he hold the autoradiograph, the X-ray film of the RFLP. Okay, so in our case, we are going to use just electrophoresis for teaching purpose. Unfortunately, I have been working since March 15 at home. Don't have the access to our own data, our students' data, which are all stored in our office computer or lab computer. So I have some hypothetical results here. And our students actually very much enjoy this experiment because they share the reagent and pipette sometime mix up. And finally, when they see and they confused the sex gene, amelogenin, why I have 2 bands. I'm a male or female, something. They're very jovial and very active in laboratory session. Here you can see our students setting up the electrophoresis. Here I have a hypothetical result like DNA profile. These are all radiograph, older version. Mother, child, male 1 and 2, and you can just compare and easily say and who is actually the father. And here I have another hypothetical result from forensic scene. Here you can see the victim DNA, victim's DNA profile, and another profile from crime scene that could be from the equipment, the weapon that they used to murder. And there are 2 suspects. There are 2 people seen from the crime scene, or they have a suspect related to the victim somehow. Okay, from the initial investigation, we also have 2 different samples. You can by naked eye you can see that the suspect one sample is exactly matched with this DNA found in the crime scene. But unfortunately, the both paternity test and the forensic test is not going to be that easy. As I mentioned, there are a lot of statistical calculation and analysis before we move on. So I need another 20 minutes, 15 minutes. So do we have a break or we just go ahead? Madam?
No, sir, we can go ahead, sir.
Okay, yeah, thank you very much.
Yeah, yeah.
Okay, so I told you, so I gave you some idea about what are DNA fingerprinting, some of the milestone. Then we see what are DNA satellites, mini and mini satellite and micro satellites, and how they are used, alleles, and I also gave you some information about CODIS, and I also gave you 3 examples of STR that we commonly use in our laboratory for the teaching purpose. Now I'm going to go a little bit further. You don't need to worry about this part because nowadays these are all done automatically. You don't need to do manually calculate all this, but you still need to understand this. Okay. Unfortunately, the code is the FBI recommend everything automation. You don't need to take the sample and prepare. When you prepare in the sample, we all know how we work in the laboratory. Okay, somebody calls you and you attend the phone, then you don't know whether you put this sample or not, whether you use the same pipette tip or you change the pipette. All these contaminations are possible. Okay, I will give you some examples later on. So the code is recommend everything is automated. FBI recommends a rapid DNA. They call it a rapid DNA system. The FBI defines the fully automated hand-free process, developing code core loci, the 20 core loci profile from reference sample to buccal swab. That means you are going to take the swab, put in the machine. That is it. You are not going to do any extraction. You are not going to do any electrophoresis. The machine will give you the profile. Okay. So then once they have the profile, they will compare with the RST profile. Remember I told you there are 14 million or 1 crore profile. Then they also text with the forensic index. That is the DNA database of only the DNA profile they collected from the forensic or crime scene. They compare, then they pass on the information to the concerned authority. Then they arrest and go for further investigation and so on. Or this is done after arresting the suspect. Then they produce all the documents to the court. So this is one of the methods, the system approved by FDA. FBI doesn't approve any methods. But they give some list. These are the things you can use. This is a fully automated system called Andy. I don't know the expansion of Andy. So it's a trademark, and they also called FluxPlex, where you can actually do 24 STR analysis. This is the swab. They just take the swab, put here in the cartridge here, just insert in the machine and switch it on. All the solution exchange of extraction buffer and the PCR buffer are fully automated. They are performed by a pneumatic pressure, and by 2 or 3 hours you have a clean profile in this monitor or transferred to the computer. Then you have the DNA data. Then goes how does you set the reaction in your machine? You are going to use multiple DNA binding fluorescent dye, blue, yellow, cyan, yellow, red, and violet, and so on, like rhodamine, FITC, TRITC, and so on. Okay, and we also know the size of the DNA, 60 to 500 bases. We know we already, because the complete sequence is available in the database now. So you know theoretically this allele has a DNA base pair from 100 to 150. When you have this specific primer, your PCR product will be from this to this. So you know, say for example, the very common amylogenin, we talked about it. There is also one more. I put okay here FGA. I think I did not save it. FGA here. Okay. So by looking at this, you can actually see the results. So this is how you look in the capillary electrophoresis, which is called electrophoregram. Okay. So you have X and Y, this amylogenin, and you have the two different allele, 21 and 25. This sample, that means the fibrinogen has 21 from one DNA copy and 26 repeat from another DNA copy from this genome of this specimen. So how are you going to analyze? So the NDIS, the National DNA Index System, they have approved certain software, Gene Mapper. All these are from Thermo Fisher. Thermo Fisher is a very big company now. They bought Invitrogen, they bought the Applied Biosystem. They recently also are going to merge with the QIAGEN. In another 2 years' time, there is no more QIAGEN. Everything under Thermo Fisher. Okay, giant. In Singapore, it is a very big company. More than 1,000 people are working on various aspects. So these software are from Gene Mapper. These are the two other software from different company. You also have a free software developed by the GenBank database, NCBI. It is equally good. Okay, approved by NDIS. You can also use for your experiment. Capillary electrophoresis is not that expensive. The prices are falling down, but we don't need it for teaching purpose. Unless you have a research laboratory, you can use it. So they are not expensive. So once you have this data, a genome frequency is calculated. That is to determine the frequency with this genotype observed among the specific population, or a Asian population, or a Chinese, or a Caucasian, and so on. So the STR allele, the FBA says that the probability of two unrelated Caucasian having the identical STR profile is 1 among 575 trillion. So it is not just looking at your profile alone. So it involves a lot of statistical analysis and a calculation. I mentioned about nowadays I have last year when I traveled to Coimbatore, and also my native is Tiruchirappalli. I studied in St. Joseph's College, Trichy, for my bachelor's degree. I did my master's in Pondicherry, did my PhD in Coimbatore, and I also worked in St. Joseph's College for 2 years before I moved here. So when I travel in Trichy and Coimbatore, I saw many laboratories, many private in vitro fertilization hospital offering people who cannot naturally conceive. They can have the test tube baby, right? So I saw many laboratories. Several years, 10 years ago in Singapore, a couple, a local Chinese and a German husband is a Caucasian. They had IVF baby, and when the baby is born, you cannot differentiate. You know, like they are very you cannot say it is an Indian or Chinese because all babies are cute and beautiful. And the features, some of the features when the baby grows up show difference, and also the blood type was different compared to the parent. So they become very suspicious, and it is one of the very big hospital. You can just Google and find. Thomson Medical Center is a very famous hospital in Singapore, and they found that, and they did the genetic testing, DNA profiling of all the IVF procedure, about 300 IVF profiles they carried out for the whole week, and they found that the egg is from the mother, actual mother, the sperm is from someone else, a stranger. So the parents sued the hospital. The case went on for a long time. I don't know the ending. I don't know. Probably they settled out of the court, because raising a child, you are the mother, but you don't know who is the father, and the emotional, leave alone the cost of raising a kid, but you see the mental agony, all the sociological issue involved. So this is a very complicated issue. This is very common. I don't know what is the stringent measures they follow. So this is another story. If you just Google, you can find many stories. I gave you one or two examples. So in the case of paternity test, again, it is not going to be a very simple. They actually calculate the paternity index. Okay, that is a ratio which, given the mother's genotype, that compares the likelihood that alleged father could produce a said child to the likelihood of the randomly chosen man, a man from specific population, paternity index. So for each gene, they calculate the paternity index, all done by the software. And by multiplying all this paternity index, you get cumulative or combined CPI, combined paternity index. And from this combined paternity index, then you calculate probability of paternity. That is the final number that you give to the court. Okay, for in the case of paternity issue, that will say there is a 99.97 or 99.95% probability. It is not always 100%. Okay, there's people say, the scientists say there could be ratio difference 97.5 to 99.99, because even though we are the offspring from the parents, our genome constantly undergo mutation because of the diet and many environmental factors every day, right? So it is always, the paternity test is not going to give the probability 100%. So I showed you how the paternity index or paternity test and forensic analysis is done with all the statistical analysis and software. Now we are going to some interesting stories, some movie time. It is now movie time. Okay, I would like to add some piece of information from DNA fingerprinting in India. I don't know how many of you know this gentleman, Dr. Lalji Singh. Enamachi? Okay, no. Dr. Lalji Singh was the pioneer. He is also considered father of DNA fingerprinting in India. When I was a student in St. Joseph's College in late 90s, early 90s, he used to be like a hero or superhero for all the biologists. He develops his own indigenous system. He's actually working on genomes of snake, where he found a region called banded kite marker BKM, a sequence which he found different from different snake. And interestingly, he also found the same homologous gene is also different in human genome. We were not using the STR those days, okay? And he started using this as for DNA fingerprinting for forensic test. And later he was the DNA fingerprinting laboratory was established in Hyderabad. He becomes the founder director, and later he involved in so many. He has a lot of application in science, nature, and cell. You can imagine a high-profile scientist with a very limited facility in those days, 30 years ago, not now. Okay, and he became the vice chancellor of Banaras Indian University, and unfortunately he died a few years ago at the age of 70. It's a huge loss for our Indian scientific community. So he is considered father of DNA fingerprinting. Very interestingly, in the 1990, for the first time, a DNA fingerprinting was used in a paternity test in India in High Court of Court. All of you just Google and this gentleman, Justice Padmanabhan, you read this justice, you don't need to listen to my talk. You don't need to go to any of the scientific research papers. This justice has explained the DNA printing technology very clearly that the layman could understand. Okay, and this is a paternity test. I don't want to go to the detail, but the defense lawyer, and of course, Lalji Singh went to the court. He actually witnessed in several cases, and he himself wrote a book on my travels into witness boxes. I think I have not read the book. I don't have access. Okay, so he went to the court, but the defense lawyer mentioned that our technology that Dr. Lalji Singh laboratory use or India uses is not a foolproof because it is not related to the Western technology where they are using all this. STR marker or ENTR marker. This is a different technology, so you should not accept it. But Justice Padmanabhan accepted the validity of the test, and the case was in favor of the mother, and they proved that he is the father of this child. Probably the child is now 30-year-old, so we don't know about all this detail. We don't need to bother about it. As a scientist, we don't need to bother about it. And unfortunately, even though we had all this DNA evidences since 1990, there are several issues in terms of because we are the democratic country and the right to privacy. As a biggest democratic country, we have the right to our own privacy. Some seem like Aadhaar card, it like went several years in the court, even providing the personal information in the card is a big issue in India. So similarly, this also went to multiple court. Ram Jethmalani was the lawyer for this defense. And finally, the government of India introduced a bill in 2016, and they made some amendment in 2018. And after that, there are many cases going on regarding this paternity test and forensic test, which is accepted as a foolproof evidence for both cases. So now I'm going to some of the very interesting case stories. I will take 10 more minutes. So I think I am taking too much time. Sorry about it. I just want make things clear for the benefit of students. So I don't know how many of you are aware of this case. This happened a very, very heart-touching case. I followed it for a long time. And those who are staying in Mumbai are very aware of that. This couple, Rajesh Talwar and Mukut Talwar, is a dentist and he is a very famous surgeon. They found their only daughter, on a one unfortunate day, they found their only daughter killed, lying on the bed with a pool of blood. And unfortunately, they don't have any clue. Unfortunately, or fortunately, this guy, their servant is also missing that day. Of course, the police came. They were all looking for the servant, the driver. They were all thinking that he was killed. This girl was, or she was A 16-year girl was killed by this driver and he escaped. Unfortunately, very interesting, the next day, this guy, the Hammond-Six body was bound on the terrace of the building. Now the case become very common. You know how things happen in a place like India, because we are like huge population. And by the time the crime scene is like visited by hundreds of people, relatives, friends, and police officers, and so many people walk around. All are lost. And the police are under severe pressure, and they need to file a case and charge it. And they made a story. I don't want to say story. I don't know how to the appropriate terminology. They said that what they said without any evidence. They said that they found Arushi on a compromising position with this driver, or they had a relationship or something. And because this couple found them in the compromising position, they killed them and left the body of this guy on the terrace, something like that. But the police could not provide any DNA evidence that if this girl was raped or molested. They could not provide any of this his DNA on her body or from the crime scene in her bedroom or any of the forensic evidences. And based on this, the case went to the CBI, and then they were acquitted from the High Court. But when they went to the High Court, I think these couples were in fact convicted. They said these couples, because being the doctor, they made a precise excision on the slate and they removed all the DNA evidences. All these are showed in the court. I think these couples are still serving the sentence. You can imagine being the only daughter, and they also lost a daughter. In case they are not the real convict, and you can imagine the pain and agony of these parents. And this story was made into a beautiful movie, Talwar. I watched this movie. This is the only movie I watched from Irfan. Unfortunately, he did a fabulous job in this movie. I loved this movie. I loved his performance. It's the only movie. I don't watch a Hindi movie because I don't speak Hindi. And this is actually, I had the subtitle. So it's a beautiful movie. When you have time, you can watch. There is another also movie made based on this story, Rahasya. I did not watch it because there is no subtitle. So this is just for your information. And this is another movie that liked by many of the 90s kids, okay, and 90s or 80s kids. I don't, for people who don't know who it is, he is a superstar in Tamil or Indian film. This is a movie directed by a very famous director, a legend called Gabe Balachandar, wherein this guy pretends like a twins in order to save his job. Okay, because the boss finds he takes a leave, telling that his mother met an accident and he goes to watch soccer or what you call a football match, where the boss also comes and see him. The next day he fires this guy, and this guy tell him that it is my brother. Because did you see whether he was having a motion? So the story goes on like that. Okay, the point is, unfortunately, the identical twins share common DNA sequence. Okay, because they come from the same zygote. Okay, they will have the same DNA profiling. So in the year 2009, the two guys, Hassan and Abbas, in Germany, were involved in 6.8 million jewel heist, jewel robbery in a bank. The police had the DNA evidence from their glove. Despite they had the evidence, they could not prosecute because they don't know, or they could not establish whether the DNA belongs to Abbas or Hassan. So they were let free because they don't want to punish the innocent. They were let free. Several years ago, at the same time, there is another story happened in Malaysia, where a drug trafficking case, okay, the guy was arrested. They had the DNA access to the DNA sample. They could not establish which of the twins were involved in the drug trafficking, because drug trafficking in countries like Singapore and Malaysia leads to death sentence. Okay, the judge mentioned that from the evidence we had, we cannot deduce that at least one of the brothers took part in the crime. We can deduce, but it has not been possible to determine which one. So they don't want to send a wrong person to gallows. They don't want to sentence a wrong person to death. So both of them let free. If both of them have the same DNA fingerprinting profile, is there any methodology that we can identify them, differentiate them? Yes, it is possible. But if you can sequence the whole genome, even though they are from the same zygote, we are eating a different food, we are exposed to the different climate, environmental factors, our DNA undergo mutation, right? If you can sequence the whole genome of the two individuals and also the genome of the sample they obtained from the bank or from the crime scene, you can establish who was the guy involved. But unfortunately, in the year 2019, whole genome sequencing was just evolving and it was very complex. Only in late 2000, all this new generation technology like IIN and BioPack and so many Illumina and so many technologies came up. Now for $300, just 5,000, 10,000 rupees, you can sequence your own genome. But 10 years ago, it was very complicated. And 30 years ago, you cannot imagine to sequence a human genome, they spent $3 billion, literally $1 per every base. Okay. There is also another technology called high resolution melting. The mutation can be also identified. This is a technology we use in our laboratory to develop for diagnostic purpose, but I don't know how far it is. This is a publication. I don't know how far this is significant in terms of sensitive, in terms of going to the paternity, the forensic or paternity with the identical twins. So I have a last question. Unfortunately, you will not be able to answer because we have a very big strain. If you don't have access to any DNA sequencing technology, you have a twins, you have a fingerprint from or access to your DNA from your the crime scene. Is there any possibility to establish who was the one involved in the crime scene or a robbery? Yes, because any clue? I think you will not be able to answer, so I'm going to give the answer myself. The fingerprint. Okay, unlike the DNA fingerprint, our actual fingerprint are different. They are not genetics. Okay, so when you are in the womb, it is influenced by different factors like nutrition, pressure, and so on. If you have access to the fingerprint, of course, many of the time they wear gloves, so it is not easy task. Okay, nowadays you can. I read a news last week in Chennai. One guy watched a YouTube video to steal. So nowadays, you know, everybody is high tech. They don't just go with a raw hand, naked hand to steal or doing any crime. That is my last slide. So these are my two of my students. Now they are my staff, our research staff, Daniel and Melissa. They helped with preparing the slides. So with this, I end my talk and I will be available for your taking your question. Thank you very much. I think I took longer time because I need to explain a little bit detail for the benefit of the students. Thank you very much.
Thank you, sir. Thank you so much, sir. That was quite informative, and also it was really awesome. And we got information from the chat box also. Everybody enjoyed your session, and they all want your PPTs. So there's a request in the Telegram group also, so to share your PPTs.
Definitely, thank you, madam. There are some slides that I have not properly acknowledged or credited. I don't have the copyright permission from some of the authors, so I also have some of the information which are the property of institution. Of course, I made it personally. It is a. So I need to make some.
So you can do some addition and then you can send it to me. Then I'll share it to the participants.
Most of them are available in the public database. So let me go through the questions. I cannot see. It is like there are so many.
So chat box was disabled. So it is now only enabled. I think now we can have the questions. It was disabled throughout your lecture. Yeah, I read a question,
Now,
sir, in the chat box. That is, can a DNA sample be genetically modified with CRISPR-Cas9 in order to disguise the genetic profile?
yeah, you can actually modify the DNA sample that you are asking about the DNA sample that you want to do your analysis, right?